Okay, so congratulations, you made it to the last talk. There'll just be a little bit of wrap up and a little game and feedback stuff after this, but this is the closing lecture. And so Ruedi is back to tell us something about biological applications of DA. Thanks, Ruedi.
Okay. So I hope you didn't just make it to this point, but also make it through this point to the next lecture. And I think I will not show a single spectrum. So, because I thought you had probably seen enough of them during this week. Okay, so this was Ben's idea, or from the organizing committee, that should talk about applications. So it's usually not super interesting if someone just comes and puts different stories behind one after the other. So I try to frame it into a common context. And we'll see how this goes. And the context is predictions. So I think we are all in one form or another in sciences. And one of the key objectives in many fields of science is to make predictions about the behavior, for instance, of a system. So this is a very simple mechanical system. And it's a nice one because we can make predictions about it. So this is, when we know the state of the system at one point, we know some things we can measure, some parameters, like the length of this string, the weight down here. We will be able to basically predict where this arrow will be at a certain time in the future, and actually even measures it directly then. So why can we do this? Because we can take this nice wooden device, we can abstract and into a specific abstraction where the relevant parts are designated, and if we have the skills to do that, we can write down some equations which basically relate these various parameters to each other and then create a formula that has a solution. And this solution allows a person to say, for instance, we can make predictions how the system behaves if we do something to that system. We could, for instance, predict using such an equation what happens if we increase the length or shorten the length of the string. And this prediction will be quite accurate, and if it is accurate and we can then do measurements and the prediction is conforming with the measurements, then we have achieved something. Basically, we have achieved some understanding of how the system behaves in some forms of state, but also how it behaves to changes of that state. So now we're not interested in clocks here, but we're interested, many of us at least, are interested in biology. And in biology, we also try to make predictions. And this is an abstraction, again, of a question or a scenario where we would like to make predictions, which is very, very common, and thousands of people work on that every day. And this is the relationship between the genomic makeup of a cell or a tissue or an animal and some form of phenotype. And of course, if we frame this in the clinical context, then this would be healthy or a person with particular disease. And so we would like to make predictions by reading out the nucleotide sequence of a genome. We might want to compare the nucleotide sequence of a genome between a person or a cohort of people who have a healthy phenotype and people who have a certain disease. And we would like to make certain predictions how variants that are detected in these genomes relate to the state over here, the health state of the person. So if you look through the literature these days, there is lots of papers like that, and they're very prominently published, particularly because we can now measure these genotypes with amazing speed and also accuracy and at a relatively low cost. So if we were in the same situation like the clockmaker or the engineer that works with this clock, we should be able to make certain predictions. We should be able to take such a genotype, and we should be able to make some changes in that phenotype, and we should be able to predict how this genotypic variant affects the phenotype over here, whether a mutation, for instance, causes a disease or does not cause a disease. So here I wrote down a few very simple questions that we should be able to answer, or to put it again in the prediction theme, predictions we should be able to make if we understood, like the clockmaker or the engineer understands the clock, we should be able to make these predictions if we understood the underlying system here. So we should be able to say what is the effect of any inherited mutation or somatic mutation on the phenotype. So we could imagine we would have some kind of computer model, and we would feed in the background wild type genome, and we would induce at a certain place a mutation, and the computer would tell us what this does. There's no computer that does that. We don't have such a model. We also do not have a model or a prediction how two or more independent mutations combine, whether they cancel each other out, whether they are independent, or whether they are somehow synergistic, cumulative. We do also not know how the same inherited mutation affects different genotypes, because we are all different. We all have different genotype. A mutation in one person might show a phenotypic effect. Exactly the same mutation in another person might show absolutely no effect, or a different one, which is common occurrence in medicine. And in fact, this is kind of the root, this question here is kind of the root of this large wave of activities in personalized or precision medicine. So we should be able to answer these questions, and we can't. When you say we, I think it's not just inability of us, for instance, our group. I think no one has a recipe how to answer these questions in general. So whenever we enter such a situation, we can of course say, well, let's just get more data and sequence some 100,000 more genomes. Eventually, we'll fall out. But we can also say maybe we lack some fundamental understanding. There's something missing why we cannot make these predictions. And when we go back in the history of biochemistry, when we would of course assume that these phenotypes are at some level an expression of biochemical processes that are going on in that person or in that cell, then we come to some really key publications, which are hallmark publications in the field of biochemistry, and they all point towards proteins. So this is, for instance, the publication from Anfinsen that said the protein function depends on the structure, so a protein sequence is not enough. Only if that sequence assumes a certain structure is a protein generally functional. This points towards the fact that we should view proteins when we analyze them with the mass spectrometer, of course, as polypeptides which we measure, but that the structure will also at some level be important. With a paper, classical paper from Lee Hartwell, and colleagues who postulated that biology is essentially modular, that if we measure a protein, that this protein very likely is not going to act as this entity that we measure, but it's going to act in the context of other molecules that form functional modules. And so we should probably be able to consider that also if we want to achieve some kind of understanding of the system. We, of course, have the relationship between gene, protein, and function. The central dogma, which everyone knows, and I think an amazing paper from Linus Pauling from the end of the 1940s, which they studied sickle cell anemia as a molecular disease. They connected mechanistically a genomic variability. It's exactly what I showed in this slide before. A specific mutation in one coding nucleotide of hemoglobin was linked to a structural difference in a protein complex, hemoglobin, tetrameric protein, supporting this modularity, and it showed that when this mutation changed the protein in its sequence, its structure, then it had a phenotypic effect. So these are landmark papers which point us towards the entity of proteins as important molecules. So now if we try to abstract and go from the classical times where people were studying, let's say, hemoglobin for 10 years to figure out what it is doing, we are now in the omics age, and we try to come up with a concept which basically moves these principles here that were elaborated over decades into the world where we operate now with large amounts of data with the techniques that we learned and discussed during this week. And so the conclusion, or what we arrived at, is what we refer to as the proteotype model, where we would say that where we define this proteotype as the state of the proteome, not just in the composition of proteins that are present in a cell, but also how they assume a modularity and basically form a superstructure that is clearly influenced by the genotype and is clearly close to the phenotype, that in terms it determines how the biochemical, it basically defines the biochemical state, which we believe affects the phenotype in health and in disease. So we would like to have a computer which would describe or simulate this prototype, that it would take into account, for instance, a point mutation over here in the genome and say, this genomic mutation affects somehow the abundance of certain proteins. It may affect how proteins change their organization into functional modules, for instance, enzyme complexes, or their partitioning into organelles, and we would then be able to relate this to phenotype. So that's kind of the picture we try to pursue. And we now are now going to discuss how we use these techniques, these DAA SWATH techniques, and of course many other techniques, to try to establish some ground truth or some findings to describe this phenotype, this prototype, and how it reacts to certain perturbations. So I mentioned on Monday that I think the currency of data, that we can generate with these DIA approaches is basically a matrix where we can do measurements across samples, and we can quite nicely quantify a rather large number of analytes, peptides, proteins, peptide modifications, and we can do this relatively quickly, and I think, as you learned, with a fairly high level of confidence. So now what I would like to discuss, how we can use these techniques, these DIA prototype measurements, to make biologically, clinically useful predictions. So I'd like to do this in 3 questions. The first would be, can we predict the response of a cell to a single disease-associated mutation? This is basically one of these 3 questions I mentioned in one of the very early slides. And I would like to test this experimentally, because right now we would not have such a model, but we would like to understand, why do we not have such a model? Why is it so complicated? Then the second thing I'm going to discuss, whether we can predict disease-associated perturbations if we do something to the proteome. For instance, we change the appearance of proteins, and this case is by interfering with the splicing machinery. I'd like to again measure or see whether we can predict the effects, and if we cannot predict the effects, we'd like to measure them at least. And then I would like to, the last point is, we'd like to predict changes in the organization of proteins that we can associate with the phenotype based on DIA data that we measure. So these are the 3 vignettes, and if maybe we don't get through all of them, but then we can stop. Okay, let's go to the first. This is work from a postdoc in our group, maybe he's here, Martin Mehnert, and it's a lot of work and extremely revealing. I think this is really a really interesting study. So the effect, what Martin attempted to do here, is to take specific mutations which are associated with the disease, particularly cancer, and measure at various levels how this mutation affects the organization of functional module and how it affects the biochemical state of the cell. We chose for that a protein kinase. The reason the type of protein kinase is really not that important for this discussion, but we chose a protein kinase because we assumed that if mutations affect the kinase activity, we will be able to measure it by phosphoproteomic footprinting, phosphoproteomic measurements. So what Martin did, he selected a number of mutations that come from the cancer genomic literature. He made mutant cell lines expressing these mutations, and then he measured the effect of these mutations on the topology of a complex around TERK2, the functional state of this complex, and the functional state of the proteotype as indicated by its state of phosphorylation. So this addresses a question I posed at the very beginning, and it addresses a very important and unresolved issue in cancer genomics, where thousands of genomes are being sequenced and tens of thousands of mutations have been found to be mutated, a mutation that happened between the background genome of patients and the tumor tissue. And when there's many of these distributions, it basically means that these are N mutations, and there's tens of thousands of them over a large cohort. Some are very frequent in a patient cohort, many are extremely infrequent, and then usually the frequent ones are referred to driver mutation, are considered basically simply of a frequency argument that they are important for the generation of disease, and the rare ones here are considered not so important and usually considered or termed passengers. So Martin took this protein kinase TERK2, and he chose this kinase because it is a protein kinase we can measure a functional readout, and also because it was already known that this kinase forms a protein complex with other proteins, and these proteins are related to ubiquitination. So this seems to be an enzyme module, a protein module, that is at the intersection of two big signaling systems, phosphorylation and ubiquitination. And so then he searched the literature for mutations that are present in this TERK2 kinase, and he found a lot of those. There's more than 80 mutations are registered on that protein itself in disease sequence databases, and he selected a few and these are simply the diseases where these mutations have been found to be associated with, and this is not readable, but it doesn't matter. It just means that there's a number of mutations have been found in this protein kinase gene and the protein, and they have associations from epidemiological or association studies with a number of diseases, particularly also cancer. Okay, then Martin selected several of these mutations, and these are the ones that we would pay a little bit of attention to. This is the kinase domain. This is other parts of the protein kinase sequence which are not related to its catalytic activity. And he selected a truncation here, 2 point mutations which are at roughly the same place as the truncation happened. So in the one case, this piece would be missing. We see this when he expresses that mutation in a cell line. This is shorter, because it misses the last piece. And then this is a activation loop mutant, which would probably affect the activity of the enzyme. A kinase dead mutant, this is an obligatory residue that is required for catalytic activity. And then a mutation proline to leucine somewhere in a region which we don't really know what it's doing. So each one of these mutations is generated by genetic engineering. He then expressed these proteins in cell lines, HEC293 cell lines, and then he started doing some measurements. So the first measurement he did was to measure the interactions of these proteins around the kinase. So when we go back here, we expect that these proteins form a module. This is basically what Lee Hartwell predicts and what Linus Pauling saw with hemoglobin. We expect that if there is mutations happening on the protein, it might affect the composition of the complex, and that's what we found. So this would be always the way we read this. This would be the proteins that with the particular mutations, kinase dead, truncation mutant, the other point mutations. And the plot here shows the abundance, the relative abundance compared to the wild type of these other subunits that are constituting this protein complex. Is a logarithmic scale, and we see that each one of these mutations, even a single amino acid point mutation somewhere in this protein, has an effect on the composition of the complex. This is remarkable, and also remarkable is that each one of these mutations has a different effect on the composition of this complex. Not surprisingly, the truncation mutant has the biggest effect, as it destabilizes the complex to the largest extent, but it does not completely disrupt it. So this is the first type of measurements. We see that the mutations affect the organization of a specific module, and different mutations affect it in different ways. So now we could also ask, since this is a kinase, we would assume that this kinase, the activity state of the kinase, is affected by its state of phosphorylation. There's oftentimes we find phosphorylations that are activating a kinase, and Martin found on the kinase number of phosphorylation sites. The blue ones were already known, and the kind of violet ones are new ones that he found. And then he could measure these phosphorylation sites, quantify them in each one of the proteins that have a mutation. And again we see, these again the mutations, these are the various phosphorylation sites. And again we see that each one of these mutations, even they have nothing to do with the actual kinase domain, they have an effect on the phosphorylation state and by inference on the activity state of this protein kinase. So now this leads us then to assume that if we were to measure the phosphoproteome in these cells, that we would likely find differences in the phosphoproteome, which would be indicating biochemical changes that are related to a phenotype. So this is what Martin did. It's actually a lot of work. He had to knock out by CRISPR from the intrinsic protein DYRK2 protein kinase. That is no extra wild type kinase activity present. And he reconstituted then these cells with any one of these mutations. This just shows that they were expressed. And then he grew up these cells. He extracted phosphopeptides. He generated a DIA SWATH data set of each one of these cell lines on the phosphoproteomic map, and then analyzed the data with the tools that you now know very well. So what came out? We see that these at the initial level, these mutations don't seem to do a whole lot, because he could quantify a fairly consistent number of phosphopeptides from these cells, and it's shown in this data matrix. But when he started to look in more detail, it turns out that each one of these mutations, probably through the organization of this protein in a functional complex that is somewhat perturbed, had a separate idiotypic and different footprint in terms of phosphoproteome in these cells. So these are the way we read this. These are proteins that are identified as phosphorylated. These would be the various mutants. They're listed down here. We have one additional one, which is a total knockout, the absence of this kinase, then the truncation, the dead mutant, and the point mutations. And each one of these mutants has a different phosphoproteomic footprint. Also in terms of number, but also in terms of proteins. Now, of course, we can make a link, because these are now not just molecules, they're molecules that carry out a function, and these proteins, and for which the function is controlled to some extent anyway, to protein phosphorylation. So what he found is that these mutations, in one way or another, each one in its own way, affect a whole range of cellular functions. For instance, epigenetic functions, transport cell migration processes, transport processes through the nuclear pore, and also cell cycle processes. So what we can conclude from this experiment is that if we make now the analogy back to the clock, where we have an equation, and we can say if we increase the length of the pendulum, we can predict what happens to the clock in terms of its state. We can certainly say here that this biology is so, the biological processes that interpret a mutation on a specific protein are so complicated that I think we're very far away from having some form of a model that would make these predictions. I think everyone would be very hard pressed to say why, from a mechanistic point of view, for instance, a mutation outside of the kinase domain in this protein affects certain biochemical functions that are different from biochemical functions generated by the mutation of the same protein in the same cell at a different site. So the conclusion would be that, this is a point I made throughout, that we presently clearly lack a model that accurately predicts the effects of a mutation on the prototype and therefore on the phenotype. So the conclusion would be that we would take from this type of study, which admittedly is a limited study with one enzyme, but I think it shows some general challenges of making a link between genetic variability or genomic variability and phenotypes, that we would be able to contribute greatly to the understanding if we can define this prototype experimentally. Okay, now I would like to come to the second question. Can we predict how a perturbation, again a disease-associated perturbation, in this case we affect the splicing machinery, we basically change the landscape of the proteins in the cell considerably by forcing the cell to express different variants of the proteins. I would like to see how this affects the organization of the proteome in terms of functional modules. So this is work that's been ongoing for quite a while in our group. We extended essentially the technique that we now learned for a whole week, like going from in a peptide-centric analysis to do protein quantification. We extended that to the level of protein complexes. So the goal is that we would like to have an experimental approach to understand how protein complexes function in the cell, which ones are present, and whether under certain conditions they change in their abundance or their composition. And we basically came up with what we call complex-centric analysis in analogy to the peptide-centric analysis. We use again prior information. This is from databases which we know that proteins interact. And we use this as prior information to inform an algorithm that would then find in a data set I'm going to describe in a second whether a specific complex that is known to exist in principle is present and detected and can be quantified in a particular sample. So the experimental side of this is certainly not a new principle. It's a very well-tested approach, a native separation of complexes, or separation of native complexes. And we use size exclusion chromatography, but Isabel, who is actually here, has been very strongly involved in this project as well as in the course, has shown that any other separation tool that maintains complexes natively will basically lead to the same result. So it's compatible with the approach. So we basically separate native complexes. We then sequence or subject each fraction, about 60 to 80 fractions, to SWATH-MS spectrometry. and we generate then a data set where thousands of proteins are basically quantified across about 60 to 80 fractions. And then we use prior information from databases to ask, for instance, this red, blue, and black complex, do we find evidence that this complex exists in this sample? This is in principle exactly the same questions you answered with transitions related to peptides. We now use peptides related to proteins and protein complexes. So there's an error model with a decoy strategy that at the end we can say this protein complex which is present in a database, for instance Quorum or Bioplex or any other database, we have evidence that allows us to say this complex was present in that sample at a certain quantity, and we have an error model that says we can make this statement with a certain probability or likelihood that this result is correct. Okay, so what can we do with that? We can measure, for instance, what fraction of the total proteome in a cell line is likely to be in complexes or is not in complexes, or at least not in complexes that are stable enough to survive this whole procedure. And this was a simple experiment from just fractionating cells from complexes from HEC293 cells. And then this analysis led to close to 5,000 proteins. So these are quantitative protein traces which are tracked through each one of these 80 fractions. And then we could measure the proteins, the proteins that are in complex because they don't migrate on this column where they would if they were monomers, or when they are in a higher molecular form. And the result was that about 58% of the proteins that are extracted from these cells appear to be bound to other proteins, at least to the degree that they move substantially differently in the column, appear much larger than they actually are. And so we would assume that they're complex, and about 42 are monomeric. Again, this doesn't mean that they're never in a complex in the cell. It just doesn't mean whenever they were in a complex, this complex didn't survive. So this is not super interesting. I mean, it's an interesting fact. But now we'd like to see how the cells behave in their organization. This was an experiment now that, again, Isabel did with a student, now postdoc, Moritz Heusel, and a master student, Max Frank. And this started a while back when we were collaborating with a group from Ashok Venkataraman in the University of Cambridge, who studied mutations that are associated with disease that affect the splicing machinery. One of these mutations is in a protein called PRPF8. It's not really important what this is, but we could then generate cells where this protein is present in wild type, or is knocked down, basically is reduced in its abundance. And we can do a certain measurement. This is work that was done a while back by Yansheng Lu when he was still here, and he did RNA sequencing of this wild type or perturbed splice form cells, and he found several thousand alternative splice forms of various kinds, and the number of those, actually quite a sizable fraction, he was able to also recapitulate and find at the proteomic level by DIA mass spectrometry. So this shows simply that by interfering in this cell with the splicing machinery, quite substantial changes happen that force the cell to express alternative proteoforms due to the interference with the splice machinery that are also detectable at the proteomic level to some extent at least. Okay, so now this is the experiment. This was to take the cells to knock down this, to generate the landscape of the RNA, to then knock down this protein factor, do sex-SWAT analysis from the extracts from the wild-type cell and the perturbed cell, and then to use this complex-centric analysis to see what happens to the organization of the proteome. And there are several questions we can ask. We can ask, how does the alternative splicing, basically expression of alternate proteoforms, affect the global proteome assembly state? How does the alternative splicing affect the assembly and abundance of specific proteins on protein complexes. And the most ambitious question, which I'm not going to explain or talk about, is how do different protein forms, which are caused by this differential splicing, how do they integrate, or whether or how they integrate in different complexes. So this is the first question. We simply asked, does this alternative splicing, or perturbation of a single gene in this complicated cell, have a noticeable and generic effect on the organization of the proteome? And this is simply done by quantifying proteins, whether they are, and to quantify the total mass of proteins that are in one or the other cell bound to complexes based on an argument that if they migrate substantially off their migration, their elution time from the sec column, where they would elute as monomers, they're considered to be bound in a complex. We don't know what complex this is, but we know they are bound to something. Okay, so this is a remarkable result. I certainly personally didn't expect that. We find, so there's again probably about 4,000 proteins were tracked through, quantified in each one of these 80 fractions. And we can now say whether they change their assembly state or whether they're more assembled or less assembled. So there's no change for about 2,700 proteins. About 1,200 proteins are more assembled, and there's some that are less assembled. So this means that by a single perturbation of a single molecule in that cell, that is part of the splicing machinery, quite substantial, thousands of proteins, more than 1,000 proteins for sure, change in some way or another their assembly state in the complexes. It's a remarkable result, and we can see, of course, which cellular functions largely are affected, and I don't really want to go into that. So then we can also ask, how do these alternative splicing affect the assembly and abundance of specific proteins? We're not really talking yet about how they integrate into complexes. We simply say, do they change their association? And we had just now a global picture and said there's quite a few changes ongoing. Now we go and ask what proteins change. And we can do this by using, so this will be now, each curve here is basically not a transition, as you now looked at all week, but it is a peptide that is from a specific protein. So this protein here would have a number of peptides that elute over chromatographic space concurrently, and they collectively identify this protein and quantify it. We see that schematically here, this protein is gone, but its same protein is now shifted. So we would say it changed its state of association, even though we may not know with whom it interacts specifically. So Isabel has then tested this and asked how many of these proteins detected change their association as a function of this perturbation in the splicing machinery. And it's a lot. It's more than 1,000 proteins, which you can see in more than one form. And this is a volcano plot where we see proteins that change in response to this perturbation, their association. Let's look at two, how we see these results. This would be always minus the perturbation, the wild type. This is plus perturbation. And we can see here that each curve is a peptide that elutes over chromatographic space and then is detected by the method, the SWATH technique that you learned this week. We see that this protein here basically is no longer present in the same place when the splicing machinery is perturbed, and conversely this protein here is also present in a complex, and this complex is no longer present. This protein is no longer in that complex when the cell is perturbed. Where these proteins go, we don't really know. Maybe they're monomeric, maybe they go to some other complex or something happens, but they clearly change their association state. Now we can also ask, how we now go from the specific protein to protein complexes, and we can ask how do this depletion of this splicing factor affect complexes. Now in these curves, in these graphs, each curve here is a specific protein that's part of a complex that is, for instance, represented in quorum or in some other database. And we will be looking for protein complex consisting of a certain number of proteins, and schematically we would see that this complex is present in the non-perturbed state, but it's gone in the perturbed state that we know that this complex has changed. And therefore, the biochemical function it carries out. We see quite a few of such complexes, which can be seen from this pie chart. There is about 340 complexes out of quorum were detected, and a fraction of those change in response to this perturbation, namely about 49, and we see this from this volcano plot, and we can now look again at the raw data. This is now a complex graph. This is a specific protein complex is involved, it's known what it does, it's involved in spliceosome biogenesis. We see that in the wild type cell, it is nicely present, consisting of these proteins which have names here, and they nicely collude. Each curve is a protein that is forming this complex, and then in the spliced form, in the splice perturbed form, this complex is completely gone. And so that means that the interference of the splicing machinery created proteoforms which did no longer allow the formation of this factor, of this molecular machine that is involved in the assembly of the spliceosome. Clearly, an important biochemical function is perturbed, and that's apparent not necessarily by the abundance of the protein or the splice form of the protein, but by the way the protein organizes into a functional complex. So the summary of this part would be, we wanted to test whether we could predict somehow or learn rules how a perturbation in the splice machinery that leads to the expression of different proteoforms, whether and how this would affect this proteotype. And I showed that the perturbation of splicing machinery induces changes in global connectivity of the proteotype, the association state of individual proteins, the abundance and composition of specific complexes, and potentially, that I didn't show, the partition of splice forms into different complexes. This is research that's ongoing. Is ongoing. So again, like in the first vignette, I would say, if the goal is to make predictions, let's say genomic predictions, how a particular mutation affects the function, the phenotype, I think we have a lot to learn, because we certainly lack a model that accurately predicts the effects of perturbation splicing, and I think we could take in by inference other biochemical or functions on the prototype and phenotype. So now, let's see what we're doing. Should I stop, or what was the...
You have 15 minutes more on the schedule. Anyway.
Ah, okay, good. I thought 4:30 is over. Okay, that's even better. At least for me, I hope also for you.
You can also vote.
Oh, let's not. Okay, so now I would like to come to the third point here. The question is, can we, and I'll show that we can measure changes in proteome organization, but it's a sizable amount of work, because we need to grow cells, we need to break them up, we need to then run per sample about 60 to 80 SWATH runs, and this, of course, is a considerable effort. Whereas if we just do DIA measurements on the sample, we can do this relatively fast. We can do now about 20 runs a day on a machine, and it's going up rapidly to much larger numbers. So the question would be, whether we can use the DIA data as they're being generated from just single-shot analysis of let's say, a clinical sample or a cell sample. When we generate this matrix I talked about, whether we could infer changes in the abundance and the composition of protein or of protein complexes. So this is now a prediction which here I said came to the conclusion that we cannot make really accurate predictions because we lack the underlying biological knowledge, and we need to do measurements here. Now I'm going to show that we can make predictions which are actually quite meaningful. So the goal is to infer variability in the organization of these modules from DIA measurements on a cohort of samples. The experimental strategy would be to quantify as many proteins as we can. Right now this is several thousand, and these numbers are going up, in a sample cohort, and then mathematically correlate protein abundance of complex components. Basically, we look for covariance of the proteins that are known to be in a complex with a phenotype. So I will explain exactly what this means in a minute. But let's first show what the study was about. The data I'm showing it's of course a generic approach, but the data I'm showing is related to prostate cancer because we've been working on that for a while. And this would be a section of a prostate from a person who had their prostate removed because the physicians found that this person has prostate cancer. Then they make slices, and then the pathologist will designate certain areas. You probably can see this with green, blue, and red. And this means something, and this means basically this is benign tissue. This would be tumor tissue by the assessment of a pathologist. And they have a grading scale of relatively moderate, light to moderate to high grade tumors, which many tumors have. So then we punched out, this is now a representation of this factual situation. We punched out, or the pathologist punched out, 1 millimeter diameter punches, basically needle biopsies from this tissue, from the benign area and from tumor areas that have different levels of tumor level. And this is referred to as Gleason score. It's not important for this discussion. And we did this initially for 39 patients. Now we have hundreds of these patients, of this data, and generated then SWATH maps from these samples. So this was 39 patients. We did run in duplicate, and from each patient we had several needle biopsies indicating tumor and benign tissue. There's a total of 210 measurements. Now we have 1,500 of these measurements. We're trying to see what they want to tell us. This is relatively, for proteomics standards, it's a relatively large experiment, but in response to or in comparison with genomics, still a small experiment. And so this is the data set. We would have about 3,800 proteins which are quantified in this data set. And the classical approach for separating these groups in the point of view of biomarker study would be to find proteins among these 3,800 which partition or help separate the groups into disease, non-disease, or different grading of disease. So it's a classical biomarker study. And now we wanted to extend this study from the abundance of proteins to the organization of the proteins in the cell by inference, not by direct measurements. And this is work from Maria Bullian, who is a postdoc in the group. And so she starts with protein quantities derived from DIA. I just showed the data set. She uses then the composition and the stoichiometry of known protein complexes. This would be, for instance, again, quorum, could be any other database. And then she computes, or she develops formalism, where she computes the covariation of proteins that are known to be in a complex across the samples. The underlying idea is simply that if two proteins are in a complex, and there is more of the complex present, or in one sample compared to the other, both of these proteins should be increased in their abundance synchronously. And if there's less of the complex, they should go synchronously. And if the complex is perturbed, let's say they're no longer in a complex, as we've seen by the SEC data, the complexes change, then they would stop going up and down synchronously. They would have a different behavior. So that's what comes out. She calculates then basically a null distribution, which is the distribution of covariance for these proteins detected based on, let's say, the normal, the benign tissue sample. Of course, these complexes are never 100% stable, so there's some variability, but she can calculate it from one cohort, let's say the benign tissue. And then she can look in the tissue, that is tumor tissue of various grades, whether the covariance of specific protein complexes from a database falls outside this null distribution is, and then of course the further out they are, the stronger this is perturbed, the stronger is the indication that biochemically this protein complex that carries out the function, that this biochemical function is perturbed. That's what we saw with Martin Mehnert's example, where he introduced a mutation into a gene, the DYRK2 kinase, and he measured that this protein kinase complex got perturbed. That he measured that with a lot of effort, and here now we try to predict this, or at least infer it from covariance analysis. So this is some results now. So this is early results, and now, but that's I think makes the point. So Maria found more than 20,000 pairs of protein complexes from this SWAT data, of pairs of proteins in that data set that are part of protein complexes. And she found several hundred that are significantly changed between the benign and the tumor samples, based on this inferred covariance. And then we can see what they are. So this is a table. I don't expect you to read it. I just want to point out what the table shows and then show one example that illustrates the conclusions. This is simply a ranking. This is the score that comes out. The higher the score, the more perturbed these two proteins here are in the tumor compared to the benign tissue. These proteins have names. They're known to form a complex, and we would say with very high probability, the highest score, these two proteins are substantially different in their formation of a complex in the tumor as opposed to the benign tissue. The scores go down, so the lower the score, the less the effect, but these are all highly significant scores. And then the numbers over here indicate the ranks, basically the weight each individual protein would have to separate tumor and control. So we see here that, for instance, this protein would have by itself quite some weight. It would be the number 4 highest ranked protein in separating tumor and control. This one would have by itself no weight, the 1000th protein, which is way down in the noise. Together they're number 1. We also have now protein complexes here, where each one of these proteins has no weight, 1200, 3800 weight to separate the groups. Together they have a very high score, which clearly means that the association of these proteins, or the inferred association, or the inferred perturbation of the association, has biological meaning. So I could say something about all of these, but I wanted to point out one, because it's very, very plausible, and it reflects known biochemistry. This is simply data-driven by this algorithm. The enzyme LDH, lactate dehydrogenase A and B, came up. So lactate dehydrogenase is an enzyme, metabolic enzyme, and neither one, the A form nor the B form, have any significant way to separate tumor and control, but together they do. So what are these proteins? It's known that LDH exists as a tetrameric protein, very similar to hemoglobin. We know that there is 2 forms, an A and a B form, again similar to hemoglobin, except that in the LDH form, the proteins can be, the tetrameric enzyme can be constituted of different proportions of the A or the B form. One is catalytically very fast, and one is catalytically slow. So if you can imagine that if you make a form that consists of 4 fast enzymes, you have a very fast enzyme, and if you have an enzyme that consists of 4 slow subunits, you have a very slow enzyme. So what is this enzyme doing? It sits here at the conversion of pyruvate into lactate, or in the that's the LDH catalytic position here. So this would be glycolysis. Eventually pyruvate is formed, and this pyruvate can be either converted to lactate by lactate dehydrogenase, or when there is not enough capacity of the enzyme, it is going to go into the TCA cycle. So it is a branching point of metabolism, and if you imagine now that you have a very fast lactate dehydrogenase, it will convert most or all of the pyruvate coming down into lactate. If it's a very slow enzyme, then the most will go in here, which has fundamentally important consequences on the energy metabolism of the cell. So the composition of this enzyme in terms of what subunits are in it have a clear and definitive and understood biochemical effect on the cell, whether the energy is converted, pyruvate is converted over here or over here. So now we can ask, I should also say, this is in the area of the cell's biochemistry where the Warburg effect happens, basically the ability of cancer cells or the propensity to use glucose, even though they would not need in the context of respiration. So now we can ask, is not the abundance of these enzymes, but the ratio a biomarker that distinguishes tumor and normal, and it is. So this would be now not the abundance as we normally see, but it's the ratio of LDH-A to LDH-B, and you can quite nicely separate normal samples and tumor. And we can also now relate this to the grading, low grade, medium, and high grade, based on the Gleason score, and we see a continuous change in the ratio, whereas the abundance of these proteins themselves is not a biomarker for this. So what we conclude is that if we can generate data matrices of sufficient quality, where you have lots of samples, reasonable number of proteins, preferably very high number of proteins, precisely measured, and if we use the information from how these proteins are organized into functional modules from prior information, we can use an inference algorithm that tells us whether certain complexes, each one with specific biochemical function, is perturbed among different conditions. And we think this is an important way to get biomarkers that are functionally quite well defined and clearly very close to the phenotype. So I started out, and now coming to the end, I started out by saying we would like to see whether we could use basically some form of computer to predict how biochemical changes or genomic changes affect the behavior of the cell, or how the cell reacts to genomic changes, single mutations, deletions, copy number variants, that we could make predictions how they affect the phenotype. And I showed two examples, Martin Mehnert's work with Dirk2, and the perturbation of the splicing machinery that make it very implausible that anytime soon we will have a model that makes such predictions in the sense of the clock that I showed first, that we say, okay, we introduce some mutation here, and what is the outcome on the system, which is a cell or a tissue. So I think we don't have this model, but we don't have a computer, but we actually have a device, and that is, of course, the cell or an organ or a tissue. Each cell knows, has evolved programs that precisely interpret any particular perturbation, genomic or otherwise, and it will mount the response. We cannot, and this response clearly is phenotypically important. Some cells will, in response to certain mutation, decide that they have to undergo apoptosis. We don't understand the mechanism, we don't understand the logic, we cannot program it in the computer, but we can do measurements. And I think what we conclude, that we have basically a computing unit that interprets these perturbations if we are able to read them out. And I would say that the proteotype model, this prototype reacts very sensitively to perturbations. I showed this with two examples. We have more of those which are equally impressive in how fine-tuned and highly differentiated the cell responds to perturbations. We think that the prototype is very close to the phenotype for reasons I also explained, because this is where the biochemistry happens. And what I try to show is that we can now, using these DIA methods, and of course also other mass spectrometric methods, with some extensions of the basic method, we can measure the prototype quite effectively, not just from the point of view of enumerating and quantifying proteins, but also proteoforms, modifications, and increasingly also how they organize into functional modules. So we think that these techniques that you now learned this week are really important in many fields of biology, because they allow us to approach now an understanding or at least some form of description which is accurate but maybe not complete of this entity, this prototype. And I think this is a very informative entity, much more so than, for instance, transcript profiles, even though transcript profiles, of course, have complete coverage of the transcriptomic space. So with that, I would like to thank all, well, actually, we'll show that first. The work I showed here is basically work from colleagues you met this week. Ludovic Chie and Pedro Navarro, who's not here, initially were instrumental in developing this SWAT technique on ABI SciEx instruments together with Steve Tate and Ron Bonner. And then others like Ben, who was very prominently in this course. Yansheng was measured several times. George, you met. And Hannes, I think he didn't met. They made contributions, very important contributions to the development of this technique. The SEX was complex analysis, complex-centric analysis. It was initially the work of Moritz Heusel and Isabel, who was here also very prominently, and others mentioned here, particularly George and Max Frank, who made is twice even, but it's only one of them. But he must be extremely productive if he shows up twice. And he was very productive as a master student who made seminal contributions. And of course, also Ben and Claudia Martelli and Matthias, they contribute to the concept and also implementation of this tool. Dirk, two projects, Martin Maynard and Maria Boulian, did computational analysis on some of the samples, and then the effects of alternative splicing is work mostly of Yansheng Lu, initially together with Ashok's group, and now in the perturbation of the system at the level of the prototype has been measured by Isabel, Max Heusel, and Max Frank again. So I would like to thank you, all participants. I'd like to thank the instructors. It's a lot of work to put a course like that together. And I said on Monday, I think it's also extremely enjoyable, certainly for me, to see this community and how they come together, discuss things for a whole week, and are willing to spend the time. I would thank to Systems X for funding and ETH for the facilities, and of course, above all, for you for active participation. And I hope you learned a lot that you can use in your own research. So I'd like to stop here. I'm happy to answer questions if there are any, and then I think there's some game or some activity.
Okay, thanks.
Yes. So we haven't done this here. You need to do this on, let's say, clinical samples. You need to, especially with metabolites, you need to be very diligent and that things don't leak out. And so you have basically to follow very rigorous protocols, and this was not done here. The samples were, some have been in the freezers for 10 years. And because there's not, it's just how clinics work, right? Someone comes in and then they generate a sample and it goes in the freezer. It's remarkable that we actually get access to these samples, but clearly there would not be good enough to also do metabolites, because metabolites might leak out or get further processed. So we didn't do that. In other cases, we have done what you suggest. We did, and we could clearly show this was in a mouse model, in a mouse cohort, where we had like a fairly large number of strains, which are genetically similar to each other, not identical, but also not as different than outbred strains. And we measured the genomic variability, could make very clear links to protein abundance, to some extent also protein complex abundance, which is complex composition, which has genetic component to it, and then to the phenotype through actually metabolic measurements when the phenotype was metabolic. So this works. You have to do measurements, of course, at various levels, but then you can link. I think on Monday I said we have these two worlds, like the biochemical world, or mechanistic world, and the big data world, and I think this is exactly what we would like to do, is combine these data sets that we generate with the backdrop of prior biochemical knowledge to make a mechanistic model that explains the links.